Hello, my Access to HE Nublets. Welcome back to another edition of my screencasts. Today we're talking infectious disease and we're looking at the chemotherapy for pathogenic infection and bacterial drug resistance. In other words, we're looking at drugs and we're looking at drugs to treat pathogens. The regular subscribers of my weekly, monthly broadcast will notice that my slide design has changed. Wow, look at this fancy stuff. I've just basically used the slide design layout suggestions thing just to make it a little bit fruitier. I hope you enjoy the effort that I've put into it. So let's move on. Here are the assessment criteria down the right hand side. We've covered most of them. Today we'll be looking at 5.1. Evaluate the use of antibiotics, antivirals and antifungal drugs as methods of treatment for pathogenic infection. It's well, what shall I say? It's fairly straightforward. As with all my lessons, I'm sure you're all thinking. So let's get straight into it. Now, the command word for this activity is evaluate. Evaluate. So you really need to put some emphasis on the success of drugs. So we're looking at how they're good, why they're limited in terms of treating pathogenic infections. We're not going to be able to look at all three in detail. So what I'm going to do is give a general introduction and then focus on one type. So in that way we can evaluate but use a particular example. And that's what I'm going to do. Now, the evaluation is covered in 4 key thoughts to try and focus your mind on what the key topics and themes you need to get across in your assignments. You might not be able to cover everything, depends on your word count, depends on how well you do your booklet and other aspects of your booklet. So, you know, bear in mind you don't have to talk about everything, all 4 of those key thoughts. The evaluation covers all 4 key thoughts, but you don't feel like you have to cover all 4 and do a wishy-washy job and not explain yourself properly. I'd much prefer quality over quantity any day. So take it and run with it how you see fit. The first thing to know, a fundamental concept, is the effective treatment of infection involves the elimination or control of the invading microorganism whilst at the same time not harming the cells of the host. What does that mean? Well, in a lab, in a test tube, it's very easy to kill a virus or a bacteria or a fungi. We've found lots of different ways to kill things, and indeed we have. In sort of Victorian times, people killed a lot of people by trialing different drugs, but they didn't have those same rigor there is today, because the art of developing an effective drug is to actually kill the pathogen without affecting the human body. So it's very easy to kill a virus, for example, but how do you get it inside your body without killing the cells of yourself whilst leaving yourself at the end of it, bing, happy and alive? That's the challenge, and so we need to keep that in mind. Now, there are different terms that we need to use in terms of drugs to treat pathogens, and the overall arching term for drugs to treat pathogens is antimicrobials. That covers microbes, and as we know from the first lesson, a microbe is a microorganism small enough that you need a microscope to see it. And all of these 4 underneath cover the different 4 types of microorganism. The difference between antibiotics and antibacterials are lab-based or naturally occurring drugs that are developed from naturally occurring means. And that's the one type that we're going to focus on throughout the session. So, have a think. When you go to the doctors, will they not give you antibiotics for a viral infection? I'm missing the why there. Obviously, I've not done the PowerPoint very well, have I? Why, when you go to the doctors, the why is there? I'm an idiot. Why? It's like I've not wrote this PowerPoint. I actually have. It's just been a while since I've used it again. So you do forgive me if you listen to this in the morning or when you drop the kids off at the pool and watching them do their swimming lessons. Why, when you go to the doctors, will they not give you antibiotics for a viral infection? Because often we've been there and, give me some drugs, give me some wonderful drugs to treat this horrendous thing inside me. And they go, nah, it's viral, go away and wait it out. So have a think. I'm going to pause the video in 3 seconds. Have a think, come up with an answer, and then skip to the next slide. Do not skip to the next slide without having a thought in your head, because if you do, you're only cheating yourself. So 3, 2, 1, and pause. Now I'm guessing half of you won't have even put a thought into your head, because all you want to do is skip through the material, and if that's you, you're an idiot. Make sure you engage and get the thought into your head. Right, my first thought is that you're you're not an idiot. The key thought is antibacterial drugs do not work against viruses because they are structured differently, and their viruses are parasites. So viruses are parasites. So there's two things there. A bacteria is very much differently structured to a virus, which we know already. And the fact is that their operation, the way that they infect us are completely different. A bacteria lives on and around us, and a virus can only work when they get inside our cells. So actually the drugs need to be completely different, and that's why a doctor will not give you an antibiotic, because a virus is inside your cell, and that's much more difficult to get. So they just don't work. Antibacterials do not work. And that comes down, like I said, to their structure and the way that they infect us. So just as a recap, here we are. Here's a virus on the right. Here's a bacteria in the middle and a fungi on the right. We've not done too much about fungi, so if you're interested, go and have a read up. I'm not using a very good coloured pen there, black on grey, but I'll change it. Now a virus is sort of acellular. It's not classed typically as alive, but is by different scientists. Depends which source you use. I don't mind what you're saying there. They generally come under the classification of pathogens, which is a microbe and a living organism. So under our terms, by all means, use virus as a living organism. A bacteria is a prokaryote, which you're known from cell biology unit and our lessons early on in this unit, and a eukaryote would be a fungi. A fungi would be a eukaryote. Got that the wrong way as well. I'm not doing very well, but it is early on in the year. This is my first video, so you will have to be a bit patient with me. I'm doing my best, and it's all for the sake of you. Now, if you think about how a drug affects these things, they have different structural features, and you do need to know these structures, particularly of the virus and the bacteria, which I'm going to go on about. Just as a quick recap, a virus has an S protein, an M protein, nucleocapsid. It has DNA inside it, a very small amount of DNA, whereas a bacteria has a pili and a cell membrane and a bacterial chromosome and a plasmid and a flagellum and blah 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 blah. If you don't understand that, please go back to the lesson on it and refresh your memory on the structure and function of a bacteria and virus. So if you've come to this slide, I'm assuming that you're all good, you know it. And so the second key thought is, in terms of evaluation of drugs, is that not all antibiotics or antibacterials work with bacteria. You can't just give penicillin for any particular type of infection, and there's a particular reason for that. Is because they all have a particular target. They all work on a specialized bit of the bacteria. So again, it's time for think time. I know students, you love thinking and thinking on yourself. Here's a picture of a bacteria on the left. You've got 1 to 7, the structures of 1 to 7. Have a think about what they are. I'll go through them in a second, just as a quick recap. But if you were designing a drug to kill this thing, what would you do generally? What would you do? Have a think about it and come up with some ideas, and then I'll go through it. So pause the video in 5, 4, 3, 2, 1. Okay, I'll come to that in a second. Be honest with yourself. Hands up who skipped forward and didn't pause and didn't engage their brain? Well, shame on you. Give yourself a smash on the head. Right. What are they? Let's go 7 to 1, which is a bit weird, but I find it a little bit better that way. 7 is the flagellum. 6 is the pili or fimbriae. They can be interchangeable. 5 would be ribosomes. 4 would be the bacterial chromosome. Sometimes I've heard it called nucleoid. 3, the cell membrane. 1, the cell wall. And 2, the capsule or slime layer. Now, if you're thinking about a drug, how can it kill these things? Well, common sense wise, you look at any of those labels and you go, right, well, it stops it working. So if it's a flagellum, let's say it stops it from being made, or it stops it from moving. The pili, it can just not make them, or it can block them, or, make them not work. Ribosomes, it can stop them making proteins and block the transcription and translation. Hint, if you don't know what that is, go back to Biomolecules lesson 1, I think it is, on my playlist, and have a look at what transcription and translation is. Number 4, you can stop the DNA from expressing itself. You can stop it from making its proteins and its other things that are made of proteins in the cell. You can punch holes in the membrane and the cell wall and slime layer. And if you were a human and you suddenly got your eyes poked out, and you got left on your own, you a good chance you might walk into a road and get flattened by a car or something. Or you chop your legs off, T-Rex comes along, chomp, you're dead. So that's the same thing, although brutally, the same principle as killing a bacteria with a drug is that if you can mutilate that thing, and we're talking it chemically, then you're going to kill the bacteria. And that's what the drugs do. So let's look at a little bit of how they do it. Down the left here, you've got some examples. So this is the site of action, so where the drug operates, and these are the type of drug that does that. Now, when we're talking about how a drug works, the real sort of posh word, if you want the really good explanation and separate yourselves from the general run-of-the-mill Joe blogs when they're searching on Google, is the mechanism of action. So if you type in what is the mechanism of action of penicillin, you're going to get a more detailed explanation rather than how does penicillin work. Well, you're probably going to get the NHS version, which is for every man and his dog. So you've got the cell wall up here on the left. These type of let's change the colour. These type of drugs will affect the cell wall. These will do the ribosomes. These will do the sort of chromosome or DNA, RNA, whatever. These will do the cell membrane, and these will do the folate synthesis, so creating folic acid, which is really important for bacteria. Now, with the exception of with folate, you can definitely relate to the cell membrane, the nuclear information, the ribosomes, and the cell wall, because we've done it before. So over here on the right, it tells you exactly, really, a summary of the way that antimicrobials work. So, like for any example, any of these that act on the cell wall on the left here will, during cell division, is inhibited. So you can look at penicillin. So you can see that it stops or damages the cell walls. What else have we got? Damaging the cell membrane. You all know about the phospholipid bilayer, so you can see there's some examples where they just rupture the cell membrane. So if you start poking holes in things, if you get a giant hole puncher and punch a hole in somebody's stomach, you're gonna kill them. Do you know what I mean? And it's the same principle. I don't know why I'm so dark this morning, but that's exactly the same principle as a bacteria and a drug to kill bacteria. Now, you do have two different types of drug I should mention for bacteria. You have bacteriostatic drugs and bactericidal drugs. Bacteriostatic drugs just basically stop them from growing and delay their infectivity, if you like, whereas bactericidal drugs kill the actual bacteria and stop them altogether. They just nuke them. All right, so both are effective, but you just need to be aware of that. And you can see the information on there, and I'll let you have a look at it. Pause the video, you can see a little bit of a diagram, which is in your Google Classrooms anyway. But that's really why, hopefully, the main point of the slide is that you can see that different drugs have a different target. So here you can see that penicillin, for example, has a target of the cell wall, but doesn't have a target of the ribosomes or the nucleic acid or the cell membrane, for example. And that's the major point. So when you're evaluating antimicrobials or antibiotics or antibacterials, you can sort of say that not all bacterials or antibacterials work with every bacteria. They're all very different because of their target. And here you've got a summary. You can see hopefully that penicillin here with a little dash over here mainly works on gram-positive bacteria, whereas let's say amoxicillin works on a grand scale of things, and that's where you're going to nuke it, which is hopefully what you're praying when you go in the doctor and your kid's ill. Just give them something to kill it all, and that's generally what they do, because we call it a wide spectrum drug. It covers a spectrum of different bacteria, whereas penicillin, you can see it's quite a narrow and specific spectrum drug, a narrow spectrum drug. And there's some examples there that you can pause the video, have a look at it, get yourself some examples, and you can fill them in into your assignments or your booklets. Now if I go back, then these categories really fall at the top here under gram-negative and gram-positive. So why, for example, would penicillin work on a gram-positive but not really on a gram-negative? Although let's take this out as an exception to the rule. So penicillin works because it works on a gram-positive bacteria. So here on the left, on the diagram on the left, you can see gram-negative and you can see gram-positive on the right. Gram-negative, if you can remember, it's a completely different structure. So it has a phospholipid bilayer followed by a layer of peptidoglycan, and you can see this sort of like puts scaffolding rods into the next layer, which is another cell membrane. So gram-negative bacteria has like a peptidoglycan sandwich. It's got two layers of phospholipids with a peptidoglycan filling, whereas a gram-positive bacteria will have only one cell membrane, and then it'll have a really thick layer of peptidoglycan. But what does that mean? Well, in a test tube or in a petri dish or in a lab, you can drop a purple stain onto a sample. This is my stain. What a lovely diagram. I amaze myself with the artistic impression sometimes. Now you can see that you can imagine this is like a fatty layer, waxy layer, and the stain doesn't stick, it washes off. So that's why we call it gram-negative. The gram stain, the purple stain, does not stick, so it tends to stay like a reticle, and you can see the gram-negative under a microscope would look like that. Whereas if you drop this purple lilac-y stain onto a gram-positive bacteria, because this peptidoglycan is really thick and it's exposed to the outer world, let's say, then the purple stain can stick, and that's why it would call gram-positive. And that's what you would see sort of thing on this side under a microscope, and to determine whether it's gram-positive or gram-negative. So that's one reason why, for example, penicillin, if we go back, would work on gram-positive but not on gram-negative. So it would work on gram-positive but not on gram. So penicillin works with this, but it doesn't work with this. And there has to be a reason. So a quick search would give you the answer. Penicillin antibiotics inhibit the formation of peptidoglycan cross-links in the cell wall. So it stops this cross-linking in the cell wall, and therefore it makes it unstable. So you can see, I'm not going to read it all out because you can read it and pause the video yourself, but you can see that penicillin under here is most effective against gram-positive, where PBP, which is up here, you can read it, is highest. Now that emphasises my key point in that not all antibacterials work with all types of bacteria. You do the maths. Now you can choose penicillin as an example if you want to, or you can go back and choose any other examples that you want if you're interested in finding out a little bit about them. But you can just tell, do they operate on gram-positive or gram-negative? You choose. But when we're talking about evaluate the effectiveness of these antimicrobials, you can see that not all antibacterials have the same effect on different types of bacteria, which is really interesting because if you took amoxicillin the advantages of that is that you're going to make sure that you kill a wide spectrum. So if it doesn't clear up, you're pretty sure that it's one of the others that's really quite specific. And that doesn't really need any diagnosis, does it? You really get amoxicillin when you go into the doctors and the doctor says, right, there you go, there's amoxicillin, nuke it, and then come back. Now, that might be great for that, but the problem is with that is that they may cause more side effects. And the reason for that is if you're nuking a lot of different types of gram-positive and gram-negative bacteria, don't forget there's lots of bacteria in your system that are doing very good jobs for you and that you need them to sort of break down food and that kind of stuff. So that's why you may have tummy upsets and diarrhea and stuff like that when you take a wide spectrum drug. And you leave opportunity for other pathogens to come in and start to colonize your system. So if you're killing the good bacteria, you're killing the stuff that's been living in you happily, nicely, doing jobs for you, yeah, this is cool. But if you kill it and you eat something again that's got, pathogens on it, you've just given them like a green pasture of your system to go ahead and start to multiply. So you are susceptible to more infections after a course of strong antibiotics like that. Now, that's not a disadvantage to a narrow spectrum drug. So if you say, let's say, penicillin, it's really only specifically targeting a certain type of bacteria. Now that's absolutely fantastic, but sometimes that might need testing, and that might lead to a delay in sending off some tests, find out what type of bacteria it is, and then specifically diagnosing or prescribing the right antibiotic. So that's why you might get wide spectrum drugs as opposed to narrow spectrum drugs. Wide spectrum drugs attack everything, but they also cause more side effects. And the benefit of that is that it takes less time. You can get prescribed it, and hopefully it clears it up. But narrow spectrum drugs are far more potent at targeting the right type of bacteria, but that does need some investigation to find the right type of drug. And again, we're evaluating the successes of different antimicrobials, don't forget. So this is one way in which we can comment on the success of antibacterials. We're not talking about antifungals or virals. Like I said at the start, we're just focusing on antibacterials. So here's that diagram just to recap on those things, and you can get a few examples. Now, the last key thought I'm going to run through is about resistance, drug resistance. Why do certain bacteria become resistant to drugs? So you've got bacteria A, let's say, working away, and you take these drugs and you kill them every time. But somehow, on some way, this bacteria has mutated, it's evolved, and suddenly this drug stops working. What's the reason for that? Why would you take a drug one time and it's the same bacteria and it kills it, and then all of a sudden we get resistance to that drug? Well, the first port of call is to think about that if we get a drug resistance, the bacteria has to change its target. It's like changing, I suppose, the locks on your house. The thing comes to mind, if you don't want your ex-partner or your ex-husband or your ex-wife or anybody that has your keys to get into your house, you don't want them in your house, change the locks, and then that stops them getting in your house. And that's what bacteria do. They change their targets so that the drugs don't work anymore. Now that happens, by luck, by chance, by changing just randomly and then hopefully getting the right shape to avoid it. But in order to change their target, they have to change their genetic information. Now that's a crucial thing. If they want to become resistant to drugs, they have to change their targets, because don't forget, the drugs have to work on their targets. Like the door analogy, if you change the locks, you can't get in. If you change the targets, a drug can't work. So first of all, I'll take you through the ways in which bacteria might change their DNA or genetic information, and then I'll say, right, what's the effect of that change? What how do they change their target as a result of changing their DNA? So let's have a look. There are 4 ways in which we can change the genetic information. When I say we, I say bacteria. I'm now a bacteria with, you can just imagine, with bit fimbriae and pili and flagellum. Posh words, but as always, it's very, we can simplify it into common language. Spontaneous resistance, conjugation, transduction, and transformation. Spontaneous resistance, don't forget, we're talking about how bacteria change their genetic information. Well, suddenly it could just change. It can change by radiation, by chemicals, or by a transcription error. That's that word again about copying genetic material. So go back to my biomolecules video if you don't get that. But basically, we can get a gene that, or genetic information that just mutates. It might change its base order, which then changes the amino acid order, which then changes the shape of the proteins. That should be key. You should have that in your knowledge. If you don't understand that, then shame on you. So if you change the amino acid order, you change the shape of the protein, and then it doesn't work. Second way is conjugation, bacterial conjugation. The plasmid can form like a space dock. So just to set this up, here you've got bacteria A with a plasmid, and here you've got bacteria with a plasmid. This has the genetic information to resist antibiotic A. So antibiotic A does not work, but antibiotic B does work. And over here you've got this bacteria, which has the plasmid, the genetic information to resist antibiotic B. So antibiotic B does not work, but antibiotic A does. So it doesn't over here, and it does over here. So this is the gene for it down here. This is the gene to resist this antibiotic. So what happens is they form a space bridge, conjugation between the pili, and the plasmid gets passed across to this bacteria. The bacteria says, "Oh, thank you very much," and then that genetic information is inserted into the chromosome, or it can be read by the plasmid, and it starts making stuff wherever it needs to make stuff. And suddenly this bacteria not only is resistant now to bacteria or antibacterial A, it's now got this resistance to both antibacterial B and A. So now it can be resistant to both types of antibacterial. So by passing on genetic information, just by actually handing it to them, then it can change the genetic code. Now, if this pili gets into the genetic code, into the bacterial chromosome, then every time this replicates, every time it goes through binary fission, you get 2 and then 4 and then 8 and then 16 and then 32, and it exponentially rises, and that's when you get very, very quick replication of bacteria, all with this antibiotic A and B, which is very important, and why you finish a course of antibiotics, to make sure that they die so that they don't have a chance to pass on information. Third is transduction. Basically, a virus can take a dump inside a bacteria and give it some genetic information, which you weren't aware. Viruses can be used to pass on genetic information to bacteria and also kill it, maybe. And then transformation is just basically the bacteria engulfs some DNA that's floating around in the body and the system of whatever organism it's in. So the idea is that it can change its DNA, isn't it? So all those 4 things enable this bacteria to change its DNA. So what does that change in DNA, and I should say that's RNA too, is it changes the target, it changes the proteins. So what does that do? If you change the target, it inactivates a drug. It can alter target, which I just said, and it can alter the permeability of a cell. So for example here, this drug used to work. All right, this drug used to work, and this cell used to have an enzyme which was, I don't know, shaped like that. But suddenly this change of DNA has now started to make an enzyme like that with an active site that fits the drug. Again, go back to the biomolecules on enzymes, and it'll tell you. Suddenly this enzyme can now start chomping and catalyzing this drug into its little bits, and it stops working. So by changing the DNA, we've changed the shape of the enzyme, which is now magically fitted into the drug, and it just dissolves it, breaks it up, whatever you want to call it, but it stops it from working. And there's an example on the right there if you're interested. The other thing we can do is change the target. So some antibiotics like streptomycin, I've had that definitely before, inhibit bacterial protein synthesis. So if you think about it, it's ribosomes, so it can change the target of the ribosomes. And we've already covered penicillin as well. So I've already talked about that. It can change the target, and there's a few examples. The third thing it can do, it can change permeability. So some antibiotics can stop the permeability, which means it can stop things from going in and out. And if you stop things from going in and out, some drugs won't work. So if you stop, let's say, a drug that acts on the genetic material from going in and out, then you're not going to help the drug act on that DNA or RNA. If you can't get in, it can't hurt you. Or if you're a bacteria, that is. If it can't get into the bacteria cell, it can't hurt you. And interestingly enough, some bacteria can develop proteins that actively pump antibiotics out of their cell. Remember, a protein pump from our cell biology days pumps things from low to high. So if a drug gets in, it can create a protein that starts pumping it back out. The sneaky little sods. And this is the fascinating world of biotics and antibiotics and drugs. There's a couple of links there for viruses if you're interested to go a bit extra and figure out how viruses evolve. Very interesting. If you're interested, piece those trimmings onto that core knowledge. But the core knowledge for us in this little section are summarised by those 4 key thoughts, really. The evaluation of the activity of drugs to treat pathogens, antimicrobials. So they don't work on the same type, so why antibiotics don't work on viruses, why antivirals don't work on every other, etc., etc. Then you'll look into the specific system, so let's say bacterials. Why don't all antibiotics treat all bacteria? And then we've got all antibacterials have different strengths and weaknesses, wide spectrum, narrow spectrum. And finally, some drugs aren't very good because the bacteria are evolving. Now, if you're thinking about virus, you can exactly think of recently the Omicron variant, and all that's done is changed its target. It's changed its S protein or its N protein. I've not really fully looked into it yet, but you can imagine that a different strain is changing its target. So I hope that's been worthwhile, and I hope that you've learned a lot, and that those are the things that I'd hope you can do as a result of this video, and I will see you in class.